New videos every day.
Life, wisdom.
Hi, I'm Dr. Colin Ross. I'm a psychiatrist based in Dallas. And I'm a psychiatrist. That means I went to medical school and I have an MD. So I studied anatomy, physiology, and worked with cancer patients and so on when I was a medical student. I don't specialize in that area currently, but I'm interested in cancer just because I'm a human being and because it's important. And I'm interested in it as an MD. So this is a little bit of a stretch outside my usual area of psychiatry. I'm studying it, learning it. I'm not really an expert yet, but I'd say I'm fairly knowledgeable. And I want to tell you about Dr. Nicholas Gonzalez, who you can find on the internet very easily, and his trophoblast theory of cancer. And John Beard is an important figure in the history of this. And the reason I want to talk about this is I think this is an incredibly important area of the cancer field that should be getting a lot of funding, a lot of attention, should really be taken seriously. You know, Susan G. Komen Foundation should be knowledgeable about it. Everybody should be looking into this and taking it very seriously. Because what we have here, I'm convinced, is a whole different model of how cancer develops from the usual, and a treatment protocol which can take somebody who's going to be dead in 3 or 6 months from pancreatic cancer and have them alive and cancer-free 15 years later. Dr. Gonzalez has got a whole series of cases like that. And that just doesn't happen in regular oncology or just because it melted away. It just doesn't happen anywhere else. So it all starts with this guy, John Beard, back in late 19th century, early 20th century. And he was a histologist. So what he did was he made literally thousands and thousands and thousands of slides of developing embryos in all kinds of different species, starting with fish. So he wasn't doing anything at all with cancer. He wasn't a physician. He was just studying how the embryo develops. And the way the embryo develops is very similar across fish, all mammals, human beings. Of course, you can't just study fish. You eventually have to take a look at human beings, which he did. But all the basic embryology was worked out in fish, eels, and species like that. So, I mean, we all know the basic facts, which is you have an egg, you have a sperm, gets fertilized, comes down the fallopian tube, and then it implants in the wall of the uterus, and then one cell becomes 2, becomes 4, becomes 8, and next thing you know, you have a baby being born. And there's no doubt about that. That's not part of the debate here. So what he did was he looked day 1, day 2, day 3, day 4, and all these 1,000 different slides, and he noticed something that is not, again, not debated. There's no disagreement in medicine at all about this, that there's a group of cells called the trophoblast cells. So each cell is a trophoblast, and they form a tissue called the trophoblast. And what the trophoblast does is, if you think about it for a second, you've got this little one-cell beginning embryo, and it's just floating around in the uterus somewhere, and it's got to implant on the wall of the uterus, otherwise it won't survive. And what the trophoblasts do is they're basically the primitive placenta. And so they can't just kind of use some super glue and glue onto the wall. They have to do something very specific. They have to invade the wall of the uterus and create a blood supply. So the blood supply of the mother and the blood supply of the baby inside the placenta are very close together. That way oxygen and food can go from the mother into the baby, and waste products can diffuse from the baby into the mother. And how all of this develops day by day by day, you know, is very well studied, and I've ordered a bunch of books and papers about it. So the trophoblast is the early form of the placenta. But what are the features of trophoblast cells? It turns out they're exactly the same as the feature of cancer cells. Trophoblast cells are very undifferentiated and primitive. They're not like very nicely defined nerve cells or muscle cells or skin cells. So they're undifferentiated. They multiply very rapidly. They're migratory. They move all over the place. They're invasive because they have to invade the wall of the uterus to form the placenta. They're angiogenic, which means they create their own blood supply, which is part of their job. they are very good at escaping and pushing away the normal regulation of the immune system. And if they couldn't do that, the mother's immune system would go, oh, there's foreign DNA in here, which is the father's half of the DNA, and they would start attacking that just like the immune system attacks a bacteria. So the trophoblast cells have to be able to kind of disguise and ward off the immune system. And they also don't clump together in the normal fashion. They tend to break apart and migrate around. So these are all the properties of cancer cells. And what Beard noticed is, looking day by day by day, about 15% of the trophoblast cells don't stay in one place and just become the placenta. They kind of break off and migrate all over, and so you can find them in the liver and the heart and the lungs and the brain. There's trophoblast cells just sitting very quietly and inactive in all the tissues of our body. So then that's an important fact that he observed and studied in these millions of slides. Then he's thinking to himself, John Beard, well, wait a minute. There's got to be some signal that switches the trophoblast from this cancer-like entity that, if it gets out of control, literally will kill the mother. So if the trophoblast doesn't settle down and become the placenta, it becomes a choriocarcinoma, which is a highly invasive, lethal cancer that literally eats its way through the wall of the uterus and kills the mother. So since the trophoblast has to stop being undifferentiated, rapidly multiplying, invasive, angiogenic, all these other properties, there's got to be some signal that switches the switch and gets the trophoblast cells to settle down and become stable, mature placenta cells and just do their function. So he's thinking to himself, I wonder what that signal is. And he notices, if I remember right, I think it's day 56 in human development, but in different species it's a different time because it's a 9-month pregnancy for humans, it's very short for other species. But there's a point in time where the trophoblast starts to transform into the stable placenta. And that's the same time, he noticed, as the primitive pancreas shows up. So he knows, hey, same time that the switch happens with the trophoblast, the pancreas is showing up and starting to secrete pancreatic enzymes. So all of this is not thinking about cancer at all. And so he studies that more, verifies that, and does some studies to see that the pancreatic enzymes that are being secreted are actually active. And so he says to himself, maybe the pancreatic enzymes are the natural signal that switches off the trophoblast. And then he says to himself, well, wait a minute, there's these trophoblast cells all over the place. And through a series of steps and research and thinking, when we go from John Beard through to David Kelly, who was a dentist in Fort Worth in the 70s and 80s, and then through to Nicholas Gonzalez, who's the current practitioner. So there's a series of steps and refinements by these three guys, Beard, Kelly, Gonzalez. But the basic idea is, wait a minute, in normal cancer physiology, normal oncology, the idea is you've got abnormal DNA. Either it's been inherited, it's already abnormal, or there's some sort of radiation or mutation happens for some reason. For whatever the cause is, you've got abnormal DNA. The abnormal DNA causes abnormal proteins, abnormal physiology, which becomes the cancer. So there may be a trigger from the environment, but basically all the action is on abnormal DNA. What's the gene for this cancer, the genes for that cancer? And what you have then in a breast cancer is you've got normal breast cancer cells that formed a normal breast, but eventually this abnormal damaged DNA starts creating breast cancer cells. So the normal breast cancer cells de-differentiate backwards. So you didn't have cancer of the breast when you're 8. At some point, your normal breast cells started to de-differentiate backwards and become rapidly multiplying, undifferentiated, invasive, migratory equals metastasizing, escaped immune surveillance, didn't clump together and stay in one place like they should. All the properties of a breast cancer cell are the same as the properties of the trophoblast. But the model is that it's normal cells dedifferentiating backwards based on abnormal DNA. The Beard-Gonzalez model is completely different. Here the idea is that cancer doesn't really arise from normal, if I just use breast cancer as an example, doesn't arise from normal breast cancer cells. What happens is it's those trophoblast cells sitting there quietly in the normal breast tissue. Now the model doesn't say exactly what it is from the environment that triggers the trophoblast, but something or some combination of things from the environment triggers these trophoblasts, so they kind of wake up, they escape normal control and surveillance, and then they start behaving exactly like trophoblast cells do. They differentiate, they're not very differentiated, they multiply rapidly, they invade. And if a trophoblast escapes containment and control in breast tissue, local biological signals in the breast tissue influence it to start evolving sort of halfway towards normal breast tissue. So it looks like a normal breast cancer cell that's dedifferentiated backwards. Actually, it's a normal trophoblast that's dedifferentiated in the direction of a breast. But if the trophoblast escapes surveillance in your brain, then the local signals there make it start to look kind of like a brain cell or liver or wherever it might be. So it's a very different model. It's not a basic genetic defect in the trophoblast cell. It's something from the environment triggers the escape from the normal quiet state. And then you've got a genetically normal trophoblast that's behaving like trophoblast cells do. So it becomes cancer. And then the next step in the logic is, well, wait a minute. If pancreatic enzymes control the trophoblast and switch off all those cancer-like properties of normal trophoblast, so it becomes the placenta, maybe we could use the same enzymes to switch off these at age 6 or 20 or 40, switch off these trophoblasts so it would be a treatment of cancer. So this is how they actually got there in a series of very logical, well-researched steps. They didn't just one day think, we should try pancreatic enzymes. And there's details to the protocol. So Gonzalez has received a lot of criticism, and a lot of the criticism is very distorted and inaccurate. And you can see that criticism on the National Cancer Institute website, which is a whole nother story. But there's a couple of things that he had to overcome. So a lot of skeptical physicians, which would be 99% of physicians, would say, well, taking pancreatic enzymes by mouth can't possibly work because they won't make it through the stomach. They'll just be digested in the stomach. So he did a bunch of research, and others did research showing, no, those enzymes can get through your stomach and get into your small intestine just fine. So then the doctors would say, well, but they're such large molecules, they can't be absorbed from your small intestine into your blood, which is totally absurd, because if those enzymes are secreted by your pancreas into your small intestine and then absorbed into your blood, that's how they work all the time anyway. But he actually did studies showing, no, not only does it make it into the duodenum or the upper small intestine, these molecules can be absorbed through your intestinal wall and get into your bloodstream just fine. So a whole series of objections have been disproven by good, solid research. And so what we have now is he's got dozens and dozens of case studies, and he's also got dozens of case studies from the George Kelly patients back in the 70s and 80s, where they've been to the Mayo Clinic, they've been to MD Anderson, they've been to somewhere big in New York or wherever. They've had a biopsy, which proves they have a cancer. They've had MRIs. So the fact that this person has a pancreatic or an ovarian cancer or lung cancer, completely proven by mainstream top oncology people. And the life expectancy with these kind of cancers, a year, maybe 6 months, maybe 2 years. And often people have metastatic cancer, so it's already spread to their liver, it's inoperable, and this is verified by the regular oncologist. And not uncommonly, they've had some surgery and some chemotherapy already. It's better if you haven't, because then the treatment takes hold better. So these are absolutely documented, no question cancer cases with horrible, horrible survival rates. And then he starts his protocol, which is it can't be just any old nutrition store pancreatic enzymes. There's some technical stuff about the types of enzymes and how they're prepared and so on. But if you have the proper enzyme package, and then there's a whole lot of nutritional supplements. And then the other thing, which sounds a little weird, is coffee enemas, which actually were a mainstream thing even into the 60s and 70s. A book called The Merck Manual described coffee enemas as standard medical practice into the 60s or 70s. And there's a rationale and a logic for that. But the core of the treatment is these pancreatic enzymes. And so he's got people with many different forms of cancer, but the best one to illustrate is cancer of the pancreas, because it's got such a horrible life expectancy. Many people die literally 3 weeks to 3 months after they're diagnosed. The number of people who are alive 5 years out with the best chemotherapy, MD Anderson, Mayo Clinic, is like literally 1% or something. So it's a death sentence. And mainstream oncologists have criticized Gonzalez, saying, well, his sample of patients isn't a random sample like a proper study. And so it's a selection bias. He just happens to have chosen the people who are going to do really well. And that's why he's got people, 5, 10, 15, 20, 30 people alive 15 years later, where on MRI their cancer's all gone away. But that makes no scientific or medical sense, because the people who are doing mainstream oncology for cancer of the pancreas don't have anybody who's alive beyond 5 years, or very, very, very rare person. But Gonzalez has got many, many alive and MRI confirmed disease-free 10, 15 years out, which cannot possibly happen just because or randomly or because he somehow selected those people. You could select 1,000 regular oncology people with cancer of the pancreas, and the number who would survive 15 years is tiny, 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 tiny. So, are there enough studies that have been done? No. Do we need more research? Yes. But just based on this large case series, we've got a very well worked out, logical, researchable rationale for how this works and why it works. We've got this collection of amazing survivors who for sure wouldn't have lived that long otherwise. So we ought to take this very, very seriously. We, the medical profession, we, American Cancer Society, we, the culture, we, the government, the federal government, ought to look at this and go, wait a minute, this is potentially so important that we've got to take it seriously. We've got to take money out of the research budget and really put some serious money into this. And when I say put money into the research budget, I mean properly conducted research. The National Cancer Institute website describes a National Cancer Institute funded study of the Gonzalez protocol, and it says actually the people on the Gonzalez protocol survived with cancer of the pancreas, survived less than the people with the regular chemotherapy. That's what you'll read on the cancer website. But that's not the actual scientific reality of what happened. Which is another book by Gonzalez. He talks about this in great detail, and he also talks about it on his website. So just a couple of basic problems with this study. The people who got the Gonzalez protocol, over half of them never signed any informed consent forms. So that violates all the basic study requirements right there. Many of these people didn't comply with the proper Gonzalez protocol, so they didn't even get the protocol. And the guy who ran the study was on the board of and held shares in the company that had made the new oncology drug that was being compared to the Gonzalez protocol. So completely biased, and it just wasn't real science study. It was just kind of a discredit, throw them in the dustbin kind of effort. So I'm talking about an actual properly designed trial that Gonzalez is directly involved in or supervising or has input in. This is something that our culture should take really, really seriously and really invest in. So thank you for listening.